This video will focus on simple cuboidal epithelium. If you watched the simple squamous epithelium video of the kidney, you'll notice that we're looking at the same tissue slide. So this is simple cuboidal epithelium. Instead of looking toward the glomerulus for that tissue, we're going to look at all of these other structures. So I moved from the 4x objective to the 10x objective. Focus. And now I want to focus, instead of the glomerulus, I want to focus on these other structures in the kidney. So let us look right here toward the middle. In this tissue of the kidney, we have a series of tubes that are kind of like straws that allow the urine that's being formed to travel through the kidney for processing. And if we were to zoom in, if we were to move from the 10x objective to the 4x objective, we want to focus on these cells in the center that have very round circular nuclei. So I've moved the 40x objective in place, and we can see that our nuclei have changed shape. Now we can see that we have all of these different tube structures. So let's pick this one right here. What we're looking at is, if you were to look at a straw and you look down a straw from the outside, let me see if I can move that a little closer to the center of the screen. If we were to look down a straw or a box of straws from the end, you would see very circular structures. So here we have a kidney tubule, and the line of that kidney tubule, here is the opening, which is the apical space of this simple cuboidal epithelium. What we see with this dark pink line here is the extracellular matrix, or the basal layer of that tissue. So here we see extracellular matrix all the way surrounding this tubule. I know that these are cuboidal in shape because the nucleus is nice and round. So here we see the edge of one cell, the edge of one cell. And the cells are cuboidal in shape, as cube-shaped as a water balloon can be. These are not perfectly square. We need to remember that these cells are almost like water balloons. So here we're going to move so that we can look at a slightly different portion of the cell and look at these beautiful cube-shaped cells. So this is simple cuboidal epithelium. So one cell, another cell, a third cell, and we can tell the difference between simple squamous has very flat, squished nuclei, simple cuboidal has round nuclei, and then simple columnar has elongated egg-shaped nuclei. So here we have a beautiful layer of simple cuboidal epithelium. If we go back to our histology guide website, we're going to look at simple cuboidal epithelium. So here is the histology guide website, and I would encourage you to experiment with this and look at all the different levels of magnification. But here we're going to focus on the 40x, and we have these beautiful layers where it's one cell in thickness in this tube, and the difference between this tube and this tube is just the angle that it was cut. So this tube right here was cut so that we can see it from the end. This tube here was cut a little bit more on an angle, so we can see a little bit more of the inside of the tube, which we call the lumen. But pay attention to the shape of the nuclei. When we see these beautiful round nuclei with a very squared off shape to the cell, this is simple cuboidal epithelium. If we go back to our model of the glomerulus, we are now looking at the tube. So here is our simple cuboidal epithelium. This is the apical space, the lumen of the tube. We have a single layer of cells, and then this red structure here is the extracellular matrix, the basement membrane, or the basal side. Nope. Sorry, y'all. So here we have the same structure, cube-shaped cells, the lumen of that tube, the inside of that tube, and then we have basement membrane on the basal side.